Looks like we're live. I think I might need the camera today. We'll see. It's moving. So I believed we finished up the animals, right? Water balance and animals. Yeah, because I think quiz is posted. All right. Do a quick check of who's here, just in case. All right. Let's get this started. These 8 a.m. classes aren't too bad, right? Once you get used to it. Just look at it this way. Once you're done, 9 o'clock, we're done with it, for the most part. All right, so we did finish up water balance in animals. So let's plug ahead to talk about water balance in plants, which is a little bit more extensive. All right, a little bit more extensive. I don't think we're going to get through this today. I'm almost guarantee we're not going to get through it today. I have an idea of where we might be able to stop. We're going to see if we can get to that point. I don't know if we'll even get through this on Friday. So this is a bigger lecture. This is a high content lecture, but it's an important lecture, because one of those things that we need to always want to talk about is what defines the species distribution. Why are species there? All right, why are species in some location? And one of these things is our species have to deal with temperature stress, they have to deal with water stress, they have to deal with salt stress, and so forth. And with plants, plants in particular, they have to deal with not only water, but they also have to deal with gas exchange and salinity of the soil and so forth. So, issue with our plants in terms of water balance is one of drying out. All right, how do they dry out? They lose water. Where's the primary source of this water loss? It's primarily coming from evaporation from the leaves and the stems. All right, so transpiration, you can say. Now, the drying out can lead to death. And there's at least 3 different ways in which death is going to come about. So the first one is just failure of the hydraulic system. All right, so you have, at some point the plant has dried out so much that the plant can no longer transport water from the roots all the way up to the leaves, or can no longer transport solutes that they're producing throughout the plant. Why would it be tissue, failure? Why would this system fail? Again, our system relies on rigidity of the plant itself. And if you lose that rigidity, you lose some of the transport. We also have possibility that air can enter the system, and air having these embolisms there can also result in this hydraulic failure. Carbon starvation is another reason why these plants will die. And again, you think about carbon starvation and you say, well, this is all about the plant not being able to take up CO2. What does that have to do with water? Well, these plants have stomata. They have the ability to open up and close these openings through which gases can be exchanged. And one way that the plants will use to limit this water loss is to close their stomata. If they close their stomata, they also prevent gas exchange. So if you prevent gas exchange, that means the plant can't get that CO2 up, can't get that CO2 that it requires to fix and assimilate and produce sugars during photosynthesis. All right, so you've kind of reached this point where, crud, I can't continue photosynthesis, and then some other bad things will happen, and we'll talk about what that is. And lastly, plants that are stressed, especially plants that are dehydrated and wilting, are going to be more susceptible to pathogens and parasites. All right, this is an indirect effect, that's what I'm going to say, because a healthy plant might be able to resist these pathogens, might be able to resist the parasites. So if they normally get it, they can survive, they can do just fine, but a stressed plant is going to be much more susceptible. Some plants are really good at living in these dry and arid areas. We call those plants xerophytes. All right, so xerophytes are the plants that are adapted to tolerate these hot and dry environments. How do they do that? Well, they have adaptations that reduce evaporation, and they usually have adaptations that aid in water acquisition. So, what are some of the adaptations that these plants might have to reduce evaporation, transpiration? Waxing coating on a leaf. All right, limits evaporation between the cells. What else? Okay, so thicker epidermis, thicker surface. All right, instead of using wax, you make it thicker. All right, same idea. All right, same idea. We're making it harder for water to leave the interior of the leaf. What are some adaptations for water acquisition? Yeah, deep taproot. Yeah, I've got this. All right. It's a taproot. Get down to where other plants cannot get. So basically, get to those limiting resources that others don't have access to. So they do kind of capture, so instead of producing a wide, expansive root system around the plant, yeah, if you kind of focus, only need the roots around your base because you funneled water, possibly. I suspect that some of those leaf orientations is due to reducing solar radiation. So we talked about like vertical leaves and stuff. So maybe it serves dual purpose. Maybe when that trait evolved, it was like a double plus for the plant, because now they don't have to spend as much energy getting those, getting their roots out and away from the plant. Colin mentioned small needle-like leaves. Yep, surface area to volume ratios definitely will help reduce evaporation. So these are adaptations that we talked about in that very first lab, in that natural history lab. So I'm not really gonna repeat them here. Just know that our plan for these xerophytes are to reduce evaporation and have adaptations to acquire what limited water is out there. So what determines water availability? Well, it's going to be determined by the soils. All right. So in these soils, our water is going to integrate between the particles of the soil itself. So you can think of the soil as just a collection of these tiny particles. They vary in size, they vary in shape, they vary in charge. All right? And our water is going to integrate into those spaces. It's from these spaces where our plant will take up the water. So our roots aren't going into the particles. Our roots themselves will be in the spaces of these of these soil particles. And the roots then are going to take up the water from those spaces. Now we have some terms associated with the amount of water that is in those soils and that it's available to those plants. First term is our field capacity. So field capacity is the amount of water that can be held by the soil. I didn't catch that. We'll leave it. Held, H-E-L-D. So it's the amount of water that can be held by the soil. All right, so our soil particles, they have all these empty spaces. How much water can fit in that space? That is the field capacity. All right, what affects the field capacity? It's basically anything that's going to affect the space. So size of the particles, shape of the particles. All right. The other aspect of this is drainage. How quickly can that water leave the space? All right, so you're kind of at an upper limit because you can add water to the soils, and then you hit an upper limit where the soils now become flooded. They're saturated. They're at their field capacity. But a well-drained soil is going to take even more water to get that, because as we're adding water at the top, so we're adding water up to at the top, we're having water draining out below it. All right, so our field capacity primarily affected by the particle size and shape, and then also we have to consider drainage. The second term, permanent wilting point, talks about the water that is unavailable to the plants because it is bound to the soil particles, and it is tightly bound. So our soil particles, as I said, they varied in shape and size. They also vary in charge, and that charge is going to latch on and hold these water molecules. If those water molecules are held onto the soil particles, then they're not going to move freely into the root of our plant. All right. So we've got field capacity, which is how much the water is in there, and then we have some water that is bound to the soil particles and can't be taken up. The difference between these two, difference between our field capacity and the permanent wilting point, represents the amount of water that is available to our plants. That's the water that the plants can take up. All right? So our soil types, we have very different types of soil. Their type tells us information about this water, about how much water is available. So let's diagram this. You can see it a little bit better. So our graph that I'm going to draw, let's see where we are. We have soil type on our x-axis. We have amount of water on our y-axis. All right? And increase the size of this. There we go. All right, so our field capacity is that water that is available in the soils themselves. So how much water the soils can hold. It's going to look something like this. Field capacity. And this is a graph based on our soil type. So what we have is sand down on that axis, or down at the lowest point of our axis. And then we can say clay is all the way up here. So sand has large particles. Clay has really small particles. Now it almost seems counterintuitive to think that larger particles have smaller amounts. It's true. All right. There's demonstrations online that you can find where they take like different sized balls in a large beaker, and then they add water to it, and they kind of estimate how much water goes in there as a demonstration of particle sizes and so forth. And it's true. When you have these large sand particles, they take up a lot of space. the particles themselves take up a lot of space, which leaves very few spaces available for our water. The other aspect of this is high drainage for our sand. So we add water and it just drains out very, very quickly. You can see this if you go to the beach. Ocean waves come up, and then as they recede, you can see like the water leave from the sand, and it leaves very quickly. All right. Clay particles are small. We're not going to see a whole lot of drainage. Why? This is an exaggeration, but we're trying to get our point across here. So this is permanent wilting point. The actual curves may be slightly different. This might not be as curved. Maybe it's a little bit more straight. All right. Maybe I diagrammed a little too much curve, but our point here is that our permanent wilting point actually increases as we go from sand to clay. So as our clay particles, they are more charged. They're going to hold on to our water more. That means there's more water that is bound to these soil particles that's unavailable to our plants themselves. So this soil is going to have a lot more water available in it, or a lot more water in it itself, but a lot of that water is unavailable. And then, what is the amount that's available to the plants? It's this space. It's the difference between these two values that tells us the amount of water that's available to plants. So if we're at sand, you can see we have a small amount of water that's available to it. Why is it? Is it because a lot of the water is bound to the soil particles? No, it's not. It's because the soils themselves don't really hold a lot of water. They drain out too quickly. Up at our clay soils, we don't have a huge amount of water available. Why is that? Is it because the water's draining out? No, not really. It's because our water is tightly bound to those soil particles. Well, plants can't get to them. All right? So even though the soil might feel damp, plants might be suffering. They might be wilting in it because they can't get to that water. Best type of soil is what would be referred to as loam, which is a mix of our various particles. So a mix of sand, mix of clay, mix of silt. All right? I will leave the specifics of our soil types to soils. All right? And those non-ag majors, if you're interested, go sign up for the course. I don't know if there's a prerequisite for that course or not, but if you're interested, something you want to get into, go take it. I hear they do a really good job at it. All right? So the plants have to deal with this. Plants have to deal with this water issue. They have to deal with the water that's draining out of the soil. They have to deal with the water that they can't get because it's bound to our soil particles. Any questions? No? Yes? All right. Let me resize. All right. So this water availability is only part of the story. The other part is salinity of the soils. All right, because our plants are going to rely on osmosis to get the water into their roots. All right. So what is osmosis? It's the movement of water down its gradient. All right. Down its water potential gradient. It's going to go from high potential to low potential. Or in other words, if we're dealing just with solute concentrations, which we are with the salts, it's going to go from an area of low salt to an area of high salt. So if our soils start to become salty, we can get a situation where the salt concentration out in the soils exceeds that of the roots, and now our water wants to naturally move out of the roots and into the soil. All right? So salinity of our soils ultimately determines how much energy the plant has to expend to get that water. All right. In our salty soils, if the soils are saltier than our roots, then the plants are going to expend energy to increase their salt concentration right in the roots themselves, right in those root hairs. Because what they're going to try to do is set up all of these gradients to get our water to naturally flow from the soils into the root hairs and then transported from cell to cell until it can get to the xylem. And it's going to utilize this just through differences in our salt concentrations. Oh, to increase salt concentration. We're going to fix that. We're going to fix this spelling. To increase. There we go. All right. So we have some plants. We have some plants that like to live in salty soils. All right. Where do we find them? Where do we find some of these plants? Where's that? Brackish environments? Maybe. Let's be a little bit more general. Because I don't want to get into aquatic plants and talk about, marine kelp and algae and stuff. Let's focus on our lands.
Coastal vegetation.
Okay, coastal areas. Yeah, coastal areas along marine environment. You have water spray coming up. That water's leaving salts in the soils. You have those plants that have to deal with it. So dune plants. All right, they've learned how to deal with this. How do they deal with it? Concentrate salts in their roots so that water can naturally flow down its concentration gradient. Other places. What are some other places?
I don't know if salt cedars fall into this category. They're not really quite coastal.
No, they're not. But why do you think they have a name, salt cedar? Okay, where did that salty environment come from? Okay, so we have deposition, all right, and I do know I'm working on our next presentation, and it's a new topic, but drying out of water, drying out of lake beds, leaves all of those minerals and salts that were in that water in that location. So those areas that used to be formerly marine, all right, when that water receded, when it evaporated, like our areas here, all right, we used to be continental shelf. We used to be underneath the water. All right, we're going to have saltier soils. You have weathering events, all right, weathering of rock. You have rainwater that's going to dissolve some of the compounds, some of the salts, or some of the compounds in the soil particles and in the rocks themselves. They're going to add salts to the soil. So even though we're far from the marine environment, we can have different soils. Now, the speed at which this develops is going to be dependent on region, all right? So climate areas. But yeah, you can have these former water, bodies of water evaporate and leaving soils. Deserts areas also tend to have more saltier soils. I should say saltier, more saltier. Saltier soils for that exact same reason. What limited water falls down, it's can be leached up from underneath, or it dissolves some rocks, and then when the water gets taken up, it leaves the salts behind. All right, questions? So it's just the salinity of the soil is just another thing we have to consider. Now with our plants, we already kind of laid out the problem. Is that we're going to dry out. Evaporation is going to happen. So our plants are going to want to conserve as much water as they can. And when they do that, they're going to prevent transpiration. They're going to prevent that water loss. But the consequence of this is that they're not exhibiting any sort of gas exchange. So how do they close these stomata? Well, some of our plants have these guard cells. All right, so these guard cells are these cells that will swell when they have water. When there's plenty of water, they swell up and they open up, kind of like a donut. All right, and that opens up the stoma where gas can go into our leaf. All right, as the plant dries out, the guard cells lose water, they start to shrink, and then it causes them to contract and close that opening. All right. Some plants put these things in pits. All right. On the leaf, we're in our different directions, so this is actually the inside. But sometimes they put it in leaf to alter the microhabitat, the microenvironment right around those guard cells to try to keep them open a little bit longer for the gas exchange. But the point here is with these guard cells, it gives the plant a way to close off that opening so that transpiration is at a minimum. But when they do that, they can't take up any CO2. They can't take it up and fix and assimilate it in the process of photosynthesis. So, how do plants handle this? Well, there's a couple different ways. There's two different ways that we'll talk about, all right? And both ways involve altering where this fixation and assimilation is going to happen. Now, these are specialized terms. I'm sure if we talk about fixation and assimilation and you ask other faculty, they might give you slightly different answers in terms of plants. When I talk about fixation, we're taking CO2 and fixing it into some organic compound. All right? Takes the CO2 from the air, we've now integrated it into some sort of organic compound. When we assimilate that CO2, that CO2 that is in the compound perhaps is now going to be integrated into some sort of metabolic pathway or metabolic cycle to create a sugar. So even though all of our pathways, I'm sure if you've had biochemistry or you will, you'll probably see some diagram of all these metabolic pathways and arrows going everywhere. And even though I'm talking about assimilation being, let's say, all up here and the rest of photosynthesis right down here, you're going to have arrows between all of those places. All right, so I'm going to recognize we have that, but with our fixation, we're going to fix it into a molecule that can be held in one place or at one time, and then when it's ready, that molecule gets shuttled over and CO2 then gets transferred into a cycle to start creating our sugars. All right? So let's talk about how they do this. The basic cycle, the most common cycle, is the C3 cycle, or the Calvin cycle. All right? In this cycle, carbon fixation and assimilation occurs all in one step. So we take CO2 out of the air, and when we fix it, it is now fixed into an organic compound that is part of the cycle that will produce the sugar. All right? So our first step of this C3, or the Calvin cycle, is to take CO2 from the air and fix it combine it with ribulose bisphosphate, RUBP, into a molecule called 3-phosphoglycerate, or PGA. So this isn't a biochemistry class. I'm not going to ask you for full names. There are some things that you do need to know, including at least these abbreviations. So this RUBP plus CO2 will produce this PGA. All right, we'll produce this PGA. That is both our fixation and our assimilation step. We take the CO2 from the air, we get it into our cycle, because at this point now, our CO2 is going to undergo transformations to get to G3P, which is itself a sugar. All right, and then that gets passed down, many more pathways. Now, this reaction, this very first reaction here, is catalyzed by the enzyme Rubisco, ribulose bisphosphate carboxylase. All right, that's what it is. This Rubisco is found in the chloroplasts of our plants. All All right, so it's where our photosynthesis is occurring. I should say, it's where our light reactions are occurring. What's notable about Rubisco is that it shows a moderate affinity for CO2. Okay, it shows a moderate affinity of CO2. And we'll come back to this idea. All right. So then, once we fixed and assimilated this CO2, all right, we've made a 3-carbon sugar, a 3-carbon molecule. But not just one. We've actually made 2. Because this ribulose bisphosphate, this RUBP, is actually a 5-carbon sugar, and that's what's in this figure. We've got 1, 2, 3, 4, 5. We've got 5 carbons. We add 1 more carbon from the CO2 to get a total of 6 carbons. But this enzyme, Rubisco, when it adds it, it splits it into 2 of these PGAs. All right, so we've got 2 of these molecules, and that's important. So we've made 2 of them. And then we get transformation. 2 of them, transformation's happening, and we get 1 of these out. 1 of these G3Ps out, leaving 1 of these other PGAs to be enter back into this pathway here to regenerate our RUBP, our ribulose bisphosphate. All right? So the way I kind of describe it here on the slide is that 1 of the PGAs that gets produced is going to be passed to our energy production steps. It's going to be passed on to the pathways that make sugars that would then be used for ATP synthesis. The other PGA is going to be held around because it's going to be transformed back into ribulose bisphosphate, or it's going to be used to regenerate ribulose bisphosphate, thus our cycle. So once we get back to this RUBP, we can now fix and assimilate another CO2 molecule. The catch here is that 1 of these PGAs gets shuttled off to make sugars and thus ATP, but then this other one, while it's getting regenerated, is going to consume energy. So that step of regeneration costs the plant energy. Where does it get this energy? It gets it from the light reactions. What are the light reactions? That's our photosystems. That's where we're capturing the sunlight. All right? We're capturing our sunlight. We're energizing the electrons. All right? We're allowing the electrons to flow down their gradient, releasing energy along the way to produce more ATP. It's that ATP that is used to regenerate this RUBP. And you can see it takes 3 of them, 3 ATP, to regenerate this RUBP once we get to this step. All right? Now, this is a simplified description. All right? This is a simplified description. What we need to know here is that in this C3 pathway, Rubisco is taking our CO2, combining it with ribulose bisphosphate into 2 of these 3-carbon molecules, these PGAs. 1 of them goes off to make energy. 1 of them gets regenerated or is used to regenerate this RUBP. Now we said that Rubisco shows a moderate affinity for CO2. Rubisco can also bind oxygen. So CO2 and oxygen are competitive for Rubisco. You can say that they're competitive inhibitors. At lower concentrations, below the affinity level of our CO2, Rubisco will now bind to oxygen, and it does so in a process called photorespiration. So photorespiration is a pathway that occurs in our C3 plants when Rubisco binds oxygen, O2, instead of CO2. All right, and this is our pathway. It's kind of showing you 2 of them. Now, we said if Rubisco works and binds CO2, it produces 2 of these PGAs. All right, 1 goes towards energy, 1 towards regeneration. When Rubisco binds oxygen, it produces PGA, but only produces 1 of them. The other molecule is a molecule that I labeled PG. which is phosphoglycolate. Yeah, phosphoglycolate. So what happens with these? Well, PGA, that one PGA, is going to be used to regenerate our UBP so that we can have our cycle. That's going to cost us energy, just like it did if Rubisco bound CO2. What about PG? What about this phosphoglycolate? Well, it's not used to produce any sort of sugars. It's not used to produce additional energy. So overall, this entire process of photorespiration is going to steal energy from our plant. It's going to consume the energy that it has already created, that it has in storage, which is a bad thing for our plants. We're at a time where our plant should be creating energy, and instead, because our CO2 levels are low, Rubisco is binding oxygen. All right. So I am going to show you kind of a graph that demonstrates when this happens. And it's going to be a graph that we will compare with our different responses. All right, so in our graph, our x-axis is CO2 concentration. All right, this will be the CO2 concentration wherever our Rubisco is. All right, so could be outside. You know, if our concentration near Rubisco is the same as out in the environment, this is what we are. If our concentration at Rubisco is on an interior of the leaf, that's where we're talking about. Our y-axis is oxygen generation. All right, and we're going to set up our 0 point right there. So that means then, at this dashed line, 0 oxygen is produced, 0 oxygen is consumed. If we're above the line, we have oxygen generation. If we're below the line, we have oxygen consumption. And why is this important? It's because when photosynthesis actually proceeds and we are successful in creating our sugars, we are producing oxygen. All right, so that's a good thing for the plant. If we're consuming oxygen, we're going through respiration. We're consuming energy. All right, and at a time when we should be making energy, that's a bad thing. That's a bad thing. All right, so what do our C3 plants look like? Our C3 plants look something like this. All right. So our rate of oxygen generation increases as we increase the CO2. But you notice that there is this area here where we are in our negative. All right? And then we hit a point where we hit our 0, and then beyond that we are in our positive. All right? So anytime we are in our positive, we have photosynthesis occurring, which is what we want. And if we are in our negatives, we have our photorespiration. That point, that 0 point, right here, has a name. This is our compensation point, our CO2 compensation point. Our CO2 compensation point. This is our break-even point. All right. So on our slide, we have CO2 compensation point. This is our CO2 concentration, at which point photosynthesis balances respiration. Now, our respiration isn't going to stop. All right. Our plant is still respiring. Our plant is still undergoing pathways to maintain themselves, to grow, and so forth. All right. But we are going to have a point where photosynthesis exceeds that respiration, and we are a net energy producer. All right? If we're down here, we are a net energy consumer. Right, so this is our break-even point. This is what I mean when we say Rubisco has a moderate affinity for CO2. All right, so that's our Rubisco. If it was a low affinity, our compensation point would be way up here. If it has a high affinity, we're going to be much closer to this crossing point. Now, what we're seeing here with our C3 plants is that these plants are going to be susceptible to carbon starvation, because there will be times when our CO2 concentration at Rubisco will fall below our compensation point. And at that point, now our carbon starvation is starting. Now our plants are going to be stressed. If they don't get out of that situation, if conditions don't improve and allow gas exchange, our plant will die. Questions? Any questions? Which one? Photorespiration. So we have our photosynthesis net energy production up here. We've got respiration occurring down here. And for the C3 plants, when we fall below that level, Rubisco will start binding oxygen and start consuming even more oxygen, more energy. All right, so let me those at home. There is our CO2 compensation point definition. These plants are susceptible to carbon starvation. Any questions? No. All right, so this is where I was hoping we would get to, and I think that is fantastic. We are going to stop here, and then on Friday we will talk about our first adaptation, which is switching to a C4 pathway, C4 cycle. All right? All right, if you have questions, feel free to ask. I did have the next quiz that got released, that was unlocked. That was a quiz over water stress in animals. Make sure you review water movement. Make sure you understand that saltwater versus freshwater fish and so forth. All right? All right. Have a good one.